Okay. I have the great privilege of facilitating this, both because Prof. Linda Becker, who's the CEO of the Desmond Tutu HIV Foundation, when I asked her what I could say to introduce her, she just said, just say that. But it's worth mentioning she has a Wikipedia page, so you'll find enough information in there. So it's a great project. The other person, panelist joining us virtually will be Mr. Tolang Senoro, and so he will be talking to us from the regulator's point of view. I've had the great privilege of working with him, and one of the wonderful things about being a scientist in this country is having a regulator that's so open to working with the scientists to help us come up with innovative ideas, in my particular case with pharmacovigilance. So without further ado, I'm going to start. But as a disclaimer, I just want to be clear. I'm a molecular biologist, so I'm bringing together the regulator, a very powerful individual, an incredible clinician, and I'm going to support it as a very box-standard scientist. But one of the things one of the advantages of doing that is that you can ask very naive questions, which often I think we don't do. But I'd also like to challenge the audience to ask some of the tough questions. Think about some of the problems that we have in diversity in clinical trials, some of the wins that we've had, lessons we can learn from, and then also maybe to finish off with some action points. So let's just get started. There we go. So when I think about one of the issues that we see from a clinical trial point of view in this country, we know that 1 in 12 individuals in South Africa will be admitted to hospital due to adverse drug reactions. That doesn't just mean from them being toxic, there's also a whole host of situations where the drugs aren't working. This is in comparison to numbers like 1 in 1,000 in countries like Iceland. So what are the problems? Why is this happening? So we all have to understand that we have this very serious disease burden relative to our gross national income. So we have a heavy burden of disease that's disproportionate to our population size. You then add, and I do hope this is going to work, and if there are any geneticists in the audience, do not judge this as high-end postgraduate genetics. It's very basic, but we imagine that Africa started with all the different genetics of the world, and then about 100,000 years ago, a small group of individuals exited Africa. If you look up at the top over here, okay, something's wrong. Sorry, can you start it again? Thanks. One of the things that we can see with that is that your blue smarties, or a very narrow version of the population, is primarily being used for clinical trials. And when we look at the next slide, hopefully it will come up in a second, when we actually then still look at that Smarties type image of the world, if it's representative in a small way of the genetic variants around the world, the number of clinical trials in Africa, to the best of my knowledge, around 14,000, predominantly done in South Africa and Egypt. And that's out of 400,000 around the world. Those figures we took from the clinical trials website a few months ago, so they may well have changed a little bit, but it just gives you an indication of how much more we need to do. Can we have those slides? Yes? Yeah, hand up. I think that either means they're coming. Great, super, thank you. Okay, so we've got this diverse genetics of Africa, and what that means is that if you look at the diversity of the African population, we have more than 25% compared to the rest of the world combined. And so one of the other things that we see with this is that the genetic variants that we see that are rare in the rest of the world are actually quite frequent in sub-Saharan Africa. So there's this massive discrepancy between what's going on where drugs are being trialed predominantly versus what is going on here. So that's for me, that's problem number 2, diverse genetics. This is the clinical trial slide that I spoke to earlier. So what do we need to do with regards to this? How can we rectify this situation? Do we need to do more clinical trials? One of the things that we think about is if there's 25% more genetic diversity, Does that mean that the cohort of individuals that we have to do have to be 25% greater? Do we need to take into account these large numbers because the population is so diverse from a genetics point of view? They spoke earlier in the previous session about a difference between cost versus price. That's something that we need to think about. Cultural diversity, logistics and infrastructure, these have been common themes that we've heard in the last couple of hours that have come through, and I'll speak to the experts on the panel more about their viewpoints on that. But one of the things that I'm really passionate about and where I come from on this is innovation. So in the absence of playing catch-up with clinical trial numbers, if that is what it is, what can we do to address the initial issue of something like adverse drug reaction? And so we try to think a lot, our team, in terms of innovative pharmacovigilance. So there are two aspects to that we quite enjoy working on. And one of the things that we've just recently done, and are wrapping up now, is looking at dispensary data from the Pico Hospital, for example, 5 million prescriptions, 5 years of adverse drug reaction reports covering a total of about 25,000 reports. And then combining that with our team, we looked at the genomic landscape, assessing some of the key ABNI genes that are responsible for drug metabolism in the body, looking at over 360 African and European alleles. And we created, the team created this pretty cool little risk matrix, weighting each of the separate issues. And we're hoping to publish this quite soon, but one of the things that we were able to do is actually start figuring out which are the drugs that are creating the biggest problem from a cost point of view and from an adverse drug reaction point of view, which are the ones that we need to be looking after. And that can maybe give us a little bit of what we've referred to as like a pharmacovigilance heads up. And then the other thing that we do a lot, and I would be remiss if I didn't do this, my director would slap me if I didn't put this up, but one of the things we think about is, we have this disadvantage of being resource limited. But being resource limited isn't all bad. It means that you actually have to be very clever about your solutions to a problem. And often that necessity breeds ideas that wouldn't necessarily have been thought of. And so one of the things that we've been working on is making liver organoids in a dish where we can replicate the genetic mutations that are prevalent in the African population and use that as a screening tool for drugs. And then also being able to set up for the first time liver cells of African origin for the first time on the continent, which are really the gold standard when it comes to drug pipeline development. So how does that really fit in with diversity of clinical trials? One of the things that we keep thinking about is how we can support clinical trials, both in the pre-clinical trial pipeline and then post, to be able to assess what problems might be coming up. And so all of this sort of centers around this idea that we want Africa to be resilient. So coming up with new and different ways about thinking about the problem might actually be something that could support clinical trial diversity. I'm going to stop there. I'm going to let Prof. Becker speak next. If you could go to your slides, please, and then we'll move on to you.
Good afternoon, everyone. A real pleasure to be here at a very interesting conference. I was unaware of the conference before this, so I'm very pleased now to see that this is happening and encouraging innovation on our continent. So I'm going to move us now to a sort of a higher level in a way in terms of looking at the macro and demographic diversity in terms of the population groups that we need to involve in our clinical research, and that this is very much the perspective of a clinical trialist. So with a nod to the geneticists and just that stunning innovation we've just heard about, this is really the bread and butter of clinical research and diversity. And I would pose that for safe, efficacious, inclusive healthcare, we really do have to think about diversity, population diversity in clinical research. And some of the reasons I think we don't include certain population groups stretch from protectionism, pragmatism and ease of clinical trial delivery, concerns for safety and efficacy, and impact on design, cost and timeline. And I'll be coming back to each one of these during my talk. Moving immediately to the consequences, though, of not increasing diversity in our trials is that we end up with health products that are used by certain populations off license because they haven't been regulated for those particular populations. And here, pediatrics really comes to mind. So about somewhere between 1/3 and 1/2 of the drugs used by children often are off license because they simply have not been tested and therefore licensed in that population. It results in the fact that some populations or age groups are denied access. Safety issues may be unrecognized or discovered late because populations aren't included. And of course, PK and pharmacodynamic differences in different people may result in different efficacious sort of outputs. And then more of a behavioral aspect is if something is not tested in a particular group or population, that population may not become aware of that particular drug or indeed adopt it during their management. And perhaps we've seen that with vaccines most recently in our most recent pandemic. So I'm going to go through a number of very specific groups that often are left out, and one can't do a talk like this without thinking about women of reproductive potential, pregnant and lactating women. And clinical trials have historically under-enrolled women globally. And this is with little recognition for the fact that females and males biologically are vastly different. We're different from a pharmacokinetic point of view in terms of weight, body fat, hormonal changes. We're different from a pharmacodynamic point of view. And we have different drug metabolism because of our milieu, whether that be hormonally driven or simply because of our makeup. And there are some very good examples. I'm an HIV clinician, and there are very good examples within the HIV world, some of which I've highlighted here that really do bring this home. And some of the reasons for this exclusion include this historical pragmatism. It's easier to enroll men. At the clinical trial site, it's always harder to enroll women who have childcare issues, who may not be able to be available when we need them at the clinical trial site. And then this notion of protectionism. We need to protect women, particularly women of childbearing age, from research rather than through research. And this is because we fear for unexpected effects in the fetus and the newborn. Of course, the thalidomide disaster of the 1950s, 60s really set us back in this regard. So here, just to show you that things are still have a way to go, this is an analysis of the US trials done between 2000 and 2020, almost 20,000 clinical trials included in the analysis, and female enrollment was around 50% here. But when you drill down, looking at the DALYs, the proportion of DALYs that females contributed, as opposed to female inclusion from a proportion point of view, you see that there are definitely some disconnects. If you follow the yellow lines, they indicate the proportion of female patients that are less than the proportion of female DALYs. So this, indicates that it's not only overall, but in particular areas of healthcare where women are being left behind. And here's another analysis looking at a number of different health areas, all the way from cardiology, psychiatry, etc., showing that again, women are represented for burden of disease, but not represented in terms of clinical trials. And most severely affected are pregnant and lactating women. So despite a global need for the use of medication during pregnancy, the research community lacks robust evidence for safety and efficacy of treatments often taken by pregnant women. And there are 3 ethical reasons why we need to rethink this. First of all, pregnant women deserve access to effective treatment. Pregnant women deserve access to safe treatment, and pregnant women deserve equitable access to trials, carrying the prospect of direct benefit. And this is the work of a group called Phases, who have been using, it's an NIH-funded project, where they've been using HIV as a specific sort of thin edge of the sword to push this issue. And I encourage you, if you're interested in this, to go and look on the internet at the phases document, quite a lot written now, to really push the issue of finding ways to get pregnant women into clinical drug trials. And of course, we're interested in drug concentration throughout pregnancy. It may be different through various trimesters. We're worried about transplacental drug transfer and drug transfer through lactation. And all of this needs investigation, not to be ignored. Historically, minors, children, have been the pharmaceutical orphans, so they have not made it into clinical trials, again because of a protectionism kind of approach, and again because it isn't easy to enroll children. There are ethico-legal difficulties that make it more difficult to run clinical trials. Numbers are smaller, and often the investment isn't there even for pharmaceuticals to develop the pediatric side of a particular product or drug. And you can see on the top the efforts by regulators, particularly the FDA now, to push the pharmaceutical industry to have a pediatric indication right from the get-go. So now there is a requirement for pharmaceuticals to explain why they are not developing a pediatric formulation as opposed to not doing it at all. And of course now we as clinical trialists need to find ways to include children in our clinical research. And if we don't, this is what happens. So hepatitis B vaccination was first licensed in 1981. It was only licensed in minors, in people under the age of 18, 10 years later. And we only really see the impact of hepatitis B vaccination once it is in fact licensed in children and can be used as part of the EPI. So this is what we want to avoid, not being able to get the good value in children as well. And in our context here in South Africa, often adolescent girls and young women are left out of clinical research, particularly in the SRH field. And this may be, for reasons, well, there are good reasons we should include them, of course. They carry a large amount of the burden when it comes to HIV, STIs, and unintended pregnancies, but unfortunately they often are left out of the clinical trials because of ethico-legal concerns, the protectionism that we are aware of, and so regulatory approval and licensure is withheld from this group. And again, here are some of the consequences. PrEP, pre-exposure prophylaxis with FTDF, Truvada, entrocitabine, was approved in 2012 by the FDA, approved by the World Health Organization, recommended in 2015. We have only just been able to get it as an indication in pregnant women in South Africa in the last year. And Descovy, its cousin, entrocitabine, tenofovir, alafenamide, also a pre-exposure prophylactic agent, is not licensed in cisgender women at all because they were not included in the phase 3 trial. And I'm very sad to say that the dapivirine ring was licensed by SAHPRA this year and is not licensed for young women under the age of 18. So this leads to exclusion of good products. What about people living with HIV, another important group? As we become aware that people living with HIV are now living normal lives, they're chronically infected, but they have manageable conditions, one needs to start asking why we are not including people living with HIV in our clinical trials, and this needs to be modified. And there's a group, the European AIDS Treatment Group, with a project called Belong, where they really are interrogating this and saying that this needs to be overturned. Again, the reasons people may be excluded are because of efficacy or safety concerns, and again, pragmatism. In a country where almost 8 million people live with HIV, I think we need to, again, examine this. And here you see COVID-19 bringing this very much a home, where of all the vaccine trials that were done with people living with HIV, very few, or in people in general, very few included people living with HIV, and where they were, numbers were very, very small. This country, in fact, had the biggest. Our Sisonke program included 40,000 people living with HIV. And we dedicated a whole clinical trial to people living with HIV. Very important that we know whether COVID vaccines work in the population of people living with HIV. And I'm pleased to say this trial was approved by SAHPRA and our ethics committees and is now fully enrolled, and we will have data on this very soon. What happens when we leave other very important populations out, such as transgender people or people who use and inject drugs or people with disabilities? And here you see the kinds of examples of clinical trials we need. The use of PrEP, for example, in people who are using feminizing hormones, a very important question. Can we put PrEP and feminizing hormones together in the same human being? And this data just coming out in 2020 to say, yes, indeed, we can. When we don't test PrEP in people, for example, who use and inject drugs, they don't know that they can use it. And so you see in the graph below, in the study below, very few people who use and inject drugs, although PrEP is an important prevention modality for them, not being aware of it because there have been so few trials of people who use and inject drugs being included in PrEP. We are making some headway though, so I want to highlight this study. It's a drug called lenacapavir, long-acting drug for both prevention and treatment in HIV. This study is underway throughout Africa now, Southern Africa, and you can see the drug design there, where we will be able to answer the question of the tenofovir alafenamide PrEP agent in this trial. But importantly, this is what I want to bring home, is we are now including 16 and 17-year-olds in the phase 3, and participants may get pregnant and breastfeed. So we are starting to make some headway in the HIV world. And here is its brother study, also being conducted in Cape Town, where you can see the populations include cisgender men, transgender women, transgender men, and gender non-binary individuals in one single trial. So, to sum things up, if we want safe, effective, and generally available health products, we are going to need well-run ethical trials that are representative of the populations in which they will be used. Those trials may cost more and take longer, but the investment will be worth it. They may need innovative designs, particularly if populations are small, and they will need consultation and partnership with the communities representative of those populations. Ethics, regulatory, researchers, community advisory boards, and recruiters will need to include the appropriate expertise. And I'll stop there. Thanks.
You can hear the difference between the way a scientist thinks of pure genetics and someone who's actually on the ground and really understands the meaning of word diversity. Tolani, can we have you now? Have we got him online?
Yes, you do have me online. I'm not sure if my slides are showing.
They are.
Oh, there we go. Wonderful,
Thank you.
Wonderful. Thank you again, Chair, for inviting SAHPRA to this very important engagement and recognizing SAHPRA as a key stakeholder in enabling the closing of the gaps that have just been highlighted by Professor Becker, and obviously harnessing the basic sciences that would inform and enable precision medicine as a part of clinical research focus in the country. Without further ado, Chair, this presentation launches from the premise that, let me just try and move my slides. I apologize. They don't seem to be moving. Oh, there we go, yes. This presentation launches from the premise that determinants of genotypic, phenotypic, and environmental, socioeconomic, sociocultural, inter-individual diversities are linked to variability that we see in patient drug safety, efficacy, and effectiveness responses. It recognizes the inherent diversity that we have in patient populations, and the assessment of which is not well served by standard approaches that lump everything together, although adjusting for that variability through random sampling at baseline still leaves a gap where you find that effectiveness is not complete, and for reasons of confounding variables that have not been accounted for. Now, to solve that problem of being precise, of not being precise in terms of how we treat patients and how we research effectiveness and efficacy and safety of drugs, a tailored approach has been encouraged where specific treatments would be for specific patient populations with defined traits that can lead to much more predictable outcomes. I will speak more on that, Chair, on the next couple of slides. Genome-wide association studies enable genome-based tailored approaches that I've just touched on. And there have been some examples of polymorphisms that have been associated with drug response phenotypes. And some of them include the link between TMP variant alleles and the risk of death with 6-mercaptopurine and methotrexate in childhood leukemia. There's been a strong association between TMP and or NUT15 variant alleles and thiopurine toxicity in cancer treatments. And there have also been just a few examples. There's also been association of loss of function alleles in hepatic enzyme genes with decreased activation of clopidogrel and leading to propensity for blood clots in that patient population, precisely because of unaccounted polymorphisms that were not thoroughly studied. And high quality biobank data sets do enable genome-wide association studies, and I'm sure a number of previous speakers have touched on this. For instance, histopathology data, disease-specific tissue collections, organ-specific tissue collections, demographics that tend to confound study outcomes if not adjusted for. So some of this information is the information that allows us to be able to build a base upon which a tailored approach to clinical research can be performed. There are a couple of examples. I zoomed in on the Human Hereditary and Health in Africa initiative, and together with other large-scale genomic projects that have developed research infrastructure, training modalities, and supportive networks for scientists in many African institutions. And these initiatives have addressed challenges of ethics, consent for data sharing, study coordination, sample shipping, data generation, and data processing, which are critical to the biobank data management value chain, and as well as sample handling. These initiatives have generated data for over 80,000 and still counting participants as a base to build on a large-scale African cohort and cover multiple diseases, that way establishing definite associations between a disease and genetic makeup. Making a case for diversity in clinical trials. Multi-regional trials indirectly capture those diversities inherent to ethnicity, and they also account for phenotype diversity. And regional trials, I would postulate, and I would put forth that should fully represent the demographic diversity in the target patient populations in order to improve external validity, in order to avoid access issues that have been mentioned before, in order to avoid other issues that have to do with off-label use, because as per the study, a certain medicine was not indicated for the population for which now it becomes necessary to expose them to. And I would also venture that trial outcomes need to be stratified by phenotype for hypothesis generation and in order to allow tailored drug safety and efficacy research and develop this area of research and its positive implications. And I'll speak to that later in terms of clinical practice, rational prescribing, and some such. And I would also add that routine pharmacogenetic sub-studies linking main study outcome phenotypes to genotype probably needed to proactively close gaps in precision medicine research. Obviously, there are cost implications there. There are clinical trial operational implications, which I'm sure Janine would have spoken to. Implications for research, we expect highly specific and sensitive clinical study endpoints. We expect inclusion and exclusion criteria to be easily motivated at the clinical trial protocol evaluation stage. We also expect larger effect sizes, which would then imply smaller sample sizes required for similar power in a differently designed trial. And we also expect less reported suspected unexpected serious adverse reactions during clinical trials. Implications for regulation. These collaborations with industry, these collaborations with scientists, bringing SAPRA into the fore will strengthen our capacity to regulate biobanks and regulate biodata management. Of course, during ethics review, increased focus will be on the Universal Declaration of Bioethics and Human Rights, as we are dealing with samples that belong to individuals whose sovereignty and agency has to be respected if we are to conduct ethical research. Less confounding variables are expected in the evidence because a lot of variables that would probably confound the study results in a normally randomly selected sample size that balances out any diversities would be the norm. And now the implications of that is that there'll be less need for clarification of evidence from the regulator to applicants, because the data set is a little bit cleaner and is less confounded by those diversities that would probably not have been accounted for at a molecular level, which is the focus of a large part of this presentation. There will be faster benefit risk review and decision-making timelines, precisely because less clarification will be needed on some of the evidence that will be submitted for registration purposes. Now, in terms of clinical practice, obviously we expect more predictable patient outcomes. We expect increased rational prescribing. We expect less unexpected ADRs because the risk benefit would have been much clearer, and the safety safety profile of the drug would have been better presented to the regulator. And we also expect increased patient satisfaction as less unexpected ADRs are experienced at the patient level, and that obviously would encourage treatment adherence. In summary, Chair, I hope I kept to my time. In summary, Chair, the value proposition of personalized medicines in clinical practice is, and I think this has been mentioned before, the ability to offer the right drug to the right patient for the right disease at the right time with the right dosage. So genetic and metabolic data will allow drugs to be tailored to patient subgroups with all the advantages that I have mentioned in the previous slide. That's the long and short of my presentation, Chair, and thank you very much for giving me this opportunity.
Thank you very much, Talang. I have a lot of questions, and I think, do we have a hard stop at 4:30? Yeah, okay, that's unfortunate. So I'm going to ask you one question, and then Talang, I'd like to ask you one, and then open up to the audience. So this is maybe not the easiest question, but your study site at Headway Research Center, what is the biggest lesson that you learned from that with regards to clinical trials? Because it's an amazing, amazing setup.
Yeah, probably in the context of this discussion, I think it is the challenge of finding truly representative participants. I think as I was listening to your talk and to Long's talk, I'm struck that I was kind of the counterfoil in a way, that without precision, we really have to be sure that we get general, because if we're going to offer this drug to everyone, we need to make sure that that drug is going to be available to everyone, and that it will be safely used as such. And so I think, when we design the trials that we do, we really want to get the individuals who are going to benefit the most to participate, and I think that probably is a challenge. So we have lots of ways to partner with community to make sure that we really make inroads into the community, and I think that partnership with community is something that we've done extraordinarily well in this region of the world in order to have people participate in our clinical trials.
And I've seen in some of the publications talking about the work that you've done, speaking about, precisely as you said, community trust is key. So Lang, if I was to ask you one question, this is really throwing you under the bus, but as the regulator, if you had to pick one aspect of clinical trial issues that you would support as the regulator, what do you think would make the biggest difference?
Just narrow it down a little bit. It's a bit broad. There are many aspects to. So,
from a logistics point of view, from a supply chain point of view, what do you think SAPRA's? What do you think would make the biggest difference in terms of making clinical trials easier to execute in the country?
Oh yes. Obviously, the biggest bugbear in terms of clinical trial logistics would be the paperwork. Yeah? It's something that we are constantly engaged with industry about in terms of how we can lessen the administrative burden on clinical trialists, so that at least they have more time to concentrate on the bread and butter of their activity. And that's always a big challenge, because at the same time you need as comprehensive a pack of information as possible, and on the other hand, obviously you do not want to burden clinical trialists with too much paperwork. So I would say it's something that we can look into and maybe try and see how other regulators are doing that without compromising patient rights, without compromising data integrity, and without compromising completeness of applications, in order for us to be able to account during the life of the study, and also to account should anything go wrong in terms of how robustly we had applied ourselves to the clinical trial approval process.
Thanks, and that really
I don't know if that
no, that makes great sense, and it also just reinforces how fantastic it is to work with a regulator that wants to take that kind of admin away, so that you can actually get down to doing what you need to do. We've got time for one or two brief questions. Is there anyone in the audience who would like to ask one? There we go. Yes.
So I guess this will be for both of you. With regards to regulation, so with the Poppy Act in South Africa, how do we share data, across the world so that we can get better drugs that are made, better genetic information that's being shared? I mean, some of that information was shared briefly, but I mean, I guess this is going to be becoming more problem when we have more genetic data that's being produced within South Africa and Africa. How do we share that to an extent that we're not just giving everything away, but also generating something that can stay within South Africa as well? Thank you.
My short answer to you would be carefully. Do you want to, I think maybe best coming from you first.
Yeah, I mean, I just want to say that already I think we've been quite sensitive about genetic information. So, certainly the regulators and the ethicists that I deal with require, for example, more precise informed consent so that at the individual level, the individual knows exactly what genetic material is being investigated or worked on, and also what kind of data is then surfacing and maybe shared. And I think part of dealing with the Poppy Act, of course, is making sure that the individual is very aware of what's going on and consents fully. And that means informed consent, right? So being very clear about what is implied. I think also we have to get much clearer about our transfer agreements, not only at a material level, and I think we've got very good about that recently when it comes to biological samples, but I think it also needs to now figure in around data transfer. So again, that we become equal partners as regards to how that data is shared. And I agree, I think we want to be collaborative because that generally will benefit globally, right? But at the same time, I think we need to be very aware of the birthright and what the implications are. So I think along with our understanding of how clinical trials develop, how better to do clinical trials, I think that whole area has to also evolve and develop. And so I think expertise growth in that area is going to be very important as we begin to participate more globally in many areas of clinical trial work.
It is 4:30. I'm going to close, but I just want to say thank you because it's probably one of the most interesting topics that people have to think about, so it was a really great question. Thank you to the panelists.